Good morning, my dear students. I welcome you all to my video class. I hope you will watch our video class and go through the book also. So, if you have any doubt, you can clarify with your staff. The topic that I am going to take today is chapter number 8, microbes in human welfare. We know that microorganisms are very tiny and which can seen only by what is called a microscope, and these microorganisms are living everywhere. They are present in water, soil, air, and the microbes are living in human being, plants as well as in animals. So most of the microorganisms are causing diseases. The microorganisms like viruses, bacteria, fungi, protozoan, virions, prions, like the different types of microorganisms are the part of the biological system. Among these, few microorganisms are causing diseases. They are more pathogenic in nature and causing diseases both in plants, animals as well as in human beings. But some of the microorganisms are useful to us. So they are playing some role or major role in our day-to-day activities. These microorganisms which are useful in different way. So in this chapter we are going to discuss about this one, the microbes that are useful for our human welfare. And some kinds of microorganisms are living as parasites, as said earlier, causing diseases. Some kinds of microorganisms can be grown in the artificial culture medium and we can produce more colonies. So when we prepare the nutritive medium in which the microbes like yeast and algae can be grown. So here the various products we can get from the microorganisms. In our day-to-day life, we are using the different food products like what is the idli and dosa, bread, yogurt. All these are actually fermented by the different kinds of microorganisms. Here in milk, actually, the bacteria are present called Lactobacillus acidophilus and Lactobacillus lactis and Streptococcus lactis. These three bacteria are commonly called as the lab. It is nothing but lactic acid bacteria. So Lactobacillus acidophilus, Lactobacillus lactis and Streptococcus lactis. These three are called lactic acid bacteria. When added in the milk, that helps to convert the milk into curd. So here the first topic, that is microbes in household products. First one is the conversion of milk into curd. So these three bacteria are playing major role for the formation of the milk into curd. And this lactic acid bacteria, when present in our intestine, especially in the digestive system, in stomach as well as in intestine, that help to prevent the growth of the other pathogenic microorganism. The lactic acid bacteria check the growth of the pathogenic microorganism and also protect our body or protect our digestive system from the other infectious microorganisms. That is an important role played by these microorganisms when the milk protein is converted into some acid. So this commonly we call this it is a lactic acid bacteria. And another one, when we add these bacteria, Lactobacillus or Lactobacillus lactis and Streptococcus in milk during formation of curd, when we add this bacteria as a starter, or it is also called as a inoculum, the process called inoculation. When these bacteria added, this helps to convert the milk into curd. In a proper temperature or in appropriate temperature, it is in plus or minus 40 degrees centigrade. That is a suitable temperature. Plus or minus, that is 40 degrees centigrade. This bacteria grow well and convert the milk into curd. And in curd, actually this curd is better than milk. Why? Because the curd actually contains more enzymes. It actually contains more enzymes when compared to the milk. And because of the different byproducts present in the curd, right, that is better than milk. So, in our daily food material, that is especially for the conversion of milk into curd, and also as I said earlier, it is the daily food, what we take bread, that is idli dosa, and in these things, these microorganisms playing an important role. And a second product, what we call this is the yogurt. It is also a kind of curd. It's called as the, it is actually produced by the bacterial fermentation of milk. Bacterial fermentation of milk. And during formation of the yogurt, the acid is produced called lactic acid. Lactic acid is produced as a by-product. And the bacteria which is involved in this yogurt formation is two bacteria playing major role. One is called Streptococcus thermophilus, another one is called Lactobacillus bulgaricus. These two bacteria, these are playing an important role. And these bacteria act on this milk protein. The name of the milk protein is called casein. The name of the milk protein is called casein. So two important food products are that is present that is in milk. Important one. One is called the milk protein called casein. Another one is the sugar called lactose. Of course, protein, minerals, vitamins, and other nutritive materials are present. But during formation of yogurt, these two microorganisms, Streptococcus thermophilus as well as Lactobacillus bulgaricus, these coagulate the milk protein called casein, and the another is a nutritive material called lactose. Lactose is nothing but the sugar. Is a sugar is a disaccharide present in milk that is converted into lactic acid. It is converted into lactic acid. So actually the flavor of the yogurt, the taste and flavor of the yogurt is due to releasing of these acetaldehyde substances. Releasing of acetaldehyde. So this is nothing but is like a curd, and these two microorganisms playing major role for the formation of the yogurt. And the third product, the third household product what we are using is another one is cheese. Actually, it is available in our place in different flavors and also different textures. Different textures. So how the cheese is produced? Here, first the milk is actually acidified. The acidity is increased. For that process, the enzyme is added called rennet enzyme. For acidification of milk, the enzyme is added called rennet. So after acidification, only the solid substances are separated. So two substances are actually produced. One is a solid material, another one is the liquid part. Only the solid material is isolated or separated. These solid materials are compressed, and the maximum of the liquid portion from that solid part is actually removed, and that is prepared as a cheese. Of course, cheese is also is widely used in our place. So for a cheese industry, the bacteria called Lactococcus and Lactobacillus Streptococcus. These three different types of microorganisms or three different types of bacteria are used for the formation of the cheese. So when this Lactococcus, Lactobacillus and Streptococcus, when added with the milk, automatically they produce, that is acidified and forming the acid. And one more, the enzyme is also added with this, that we call this as the rennet. So cheese is also a commonly used the household product that we are getting because of these three different microorganisms. And fourth one is the paneer. Actually, it is in it is also called as a cottage cheese. This paneer is prepared from the fresh milk. prepared from fresh milk. It is a kind of a fresh cottage cheese. Paneer is the more common. It is produced in South Asia and especially in India. It is produced or used widely used in South Asia, but especially in India. So this in paneer formation or during this paneer production, this process done called curdling of milk. Curdling in the sense that is the milk is actually converted into curd not by using the microorganism, not by using the fungus or bacteria. This milk is converted into curd by adding either lemon juice or vinegar or some organic acids also. Some organic acids also used for conversion of this milk into curd-like things. But it is actually fresh one. But in other process during cheese formation or yogurt formation, in proper temperature, the milk should be fermented for a few hours, 8 to 12 hours. But here, this actually we can prepare the paneer in short duration. So fresh milk that is used, fresh product that we can get immediately. But here there is no role of any microorganisms. But here the another one is called as the Swiss cheese. It is a special type of one because the Swiss cheese is fermented by a bacterium called Propionibacterium shermani. That is name of the bacteria called Propionibacterium shermani. And the special characteristics in the Swiss cheese is large holes. That is, large holes are formed in the Swiss cheese. That is because of releasing of more carbon dioxide. When carbon dioxide is released, during a fermentation process, which creates more or large holes. So here the bacteria which is involved for formation of the Swiss cheese is Propionibacterium shermani. Is another product that is that we are using, and a duff that what we are using for making idlis and dosa. Okay, here suppose if you prepare this duff without adding any kinds of microorganisms, in the next day we can see the fermented duff. The fermentation process is actually done by the bacteria called Leuconostoc mesenteroids. That is the name of the bacteria called Leuconostoc mesenteroids. Both in bread making industry as well as in household preparation of this idli and dosa, the glucose which are present in the rice, this glucose is converted into this ethyl alcohol after fermentation process. So formation of this ethyl alcohol as well as the production of more carbon dioxide during fermentation process and a unique characteristics that is in both the industry. One is in the in household product that is produced called idli and dosa. Another one is in bread making, that is bakeries. So the Leuconostoc mesenteroids that is responsible for conversion of or that is for fermentation process. So the duff that is suitable one is prepared for useful to prepare our daily food. And next one, that is the bread making industries or in the bakeries. In bakeries we can get different types of bread that is also in different taste. But here the different taste and also different types of this bread is prepared by using this Saccharomyces cerevisiae. Saccharomyces cerevisiae is an yeast. It is an yeast. So it is commonly called a baker's yeast. Saccharomyces cerevisiae is a baker's yeast. But in other product we are using only the bacteria for the different process. But here in this case we cannot use viruses or virions or prions for preparing the different kinds of these food materials, because viruses cannot grow in non-living things. So these bacteria and yeast can be grow as these heterotrophic organisms can grow, and we can get these beneficial products. So Saccharomyces cerevisiae is also called as the baker's yeast, and when the duff is actually prepared, here the two products are that is produced. One is called ethyl alcohol and carbon dioxide. When ethyl alcohol carbon dioxide is produced, right, the bread or the product is become soft. That is called leavened duff. So when ethyl alcohol and CO2 is processed, that is called a fermented one. It is nothing but a fermented one is called a leavened duff. And when this leavened duff is that is baked in the sense when the leavened duff is actually cooked in the sense by using more heat, the leavened duff is actually baked, that releases this ethyl alcohol and also carbon dioxide. Of course, when the duff is prepared, ethyl alcohol carbon dioxide is produced, that is present in the leavened duff. When the leavened duff is baked, this ethyl alcohol and also carbon dioxide evaporates. When these two evaporates, that actually produces more holes in bread. Right? Because of these holes, the bread is become soft and also porous. The bread become soft and porous because of the evaporation of ethyl alcohol and carbon dioxide through or by making more number of holes. And also the duff during baking, the duff also slightly raises. That is also because of the carbon dioxide. So this is another household product. So if you see this, the major microorganisms that is playing the formation of household product. Here in the conversion of milk, these three: Lactobacillus acidophilus, Lactobacillus lactis, and Streptococcus lactis. For yogurt formation, that is Streptococcus thermophilus and Lactobacillus bulgaricus. And for cheese industry, these three: Lactococcus and Lactobacillus and Streptococcus. The coccus, bacillus, these are the different type of bacteria classified based on its structures. And for paneer or cottage cheese industry, as I said earlier, here there is no role of any microorganisms. Only curding of milk is done by adding either lemon juice or vinegar or some organic acids. But for formation of the Swiss cheese, the bacteria called Propionibacterium shermani, and for making this Italian dosa, Leuconostoc mesenterides, and in bread making industry, Saccharomyces cerevisiae. So these microorganisms, right, are not causing disease, but they are directly useful to human beings. Another one is called spirulina. The spirulina is nothing but a unicellular or single-celled. This is a single-celled or unicellular, that is algae. Spirulina is a unicellular algae. From this spirulina, the protein is obtained, that is called single cell protein. It is called SCP. SCP is nothing but single cell protein. So this spirulina, either in the fresh form or these are mixed with some other organisms. The special type of protein is prepared. Actually, spirulina contains more protein. This protein is added as an ingredient with the food products. The food products are prepared for human consumption, or we can prepare for the animal feed. For animal feed preparation or for human consumption. Right? This spirulina is actually used. So the protein which is obtained from spirulina is used for this animal feed preparation or for human consumption. So this is another these microorganisms which are used as food materials. Right? Okay, second one is microbes in industrial products. In industry, there are different types of products that we are obtaining. like vaccines, enzymes, different types of drugs. What we call this is pharmaceutical drugs, steroids, etc. These are actually obtained from the different industries. All these are the valuable industrial products that we are obtaining by using these microorganisms. Here, for the production of large scale industrial products, we have to use the product for in the large closed vessels. The large closed vessels, what we call is the fermenters. It's called as the fermenters. And within the fermenters, the microbes are used. The microbes are used. The microbes are called fermenters. It's called fermenters. The large closed vessel in which the fermentation process actually takes place, that is called as the fermenters, and the microbes which are used for the fermentation process called fermenters. And here in the fermenters, or it is also called bioreactors. It is also called bioreactors. In this, the proper the facilities or all the necessary facilities are made, especially for aeration, agitation, and temperature as well as pH control, and in addition to that, the vent for overflow and as well as for draining. For all these things, all facilities are made in fermenters. After fermentation process, actually the product which is produced is separated and purified to get the various the products for human use. So here the product which is produced that we are going to discuss, that is antibiotic production. Antibiotic productions. So what are called antibiotics? Antibiotics are nothing but these are chemical substances. These are chemical substances produced from one microorganism, one microorganism, which the chemical produced by one microorganism which inhibits or detrimentation and prevent the growth of the another kind of microorganisms is called as the antibiotics. Antibiotics are nothing but it is a biological product. It is a chemical substances produced by one microorganism and inhibits the growth of the another kinds of microorganisms, which is called as the antibiotics. The antibiotics are actually broadly classified into two types or in two classifications. One is called narrow spectrum antibiotics. Another one is called broad spectrum antibiotics. The narrow spectrum antibiotics can kill or destroy only the unique or specific type of microorganism. But the broad spectrum antibiotics that are able to kill or destroy the wide range of or different types of the microorganisms. This is one type of classification. Now another type of classification, generally the antibiotics are called are classified as bacteriostatic and bactericidal. The two different types of antibiotics. Bacteriostatic antibiotics generally prevent or inhibits the growth of the bacteria, but the bactericidal antibiotics kills the bacteria. It kills the cidal, C-A-D-A. The cidal meaning is actually kills. So bactericidal antibiotic kills the bacteria. Bacteriostatic antibiotic inhibits the growth of the bacteria. So here the antibiotic word, the antibiotic term antibiotic, that is actually given by the person is called Selman Waxman. His name is called Selman Waxman in the year 1943. Actually, this man Waxman discovered the antibiotic called streptomycin. Discovered the vaccine called streptomycin vaccine. The term antibiotic used by Selman Waxman in the year 1943. And actually the first antibiotic discovered by Alexander Fleming in the year 1926. Alexander Fleming discovered the antibiotic in the year 1926. Actually, when he was working in his laboratory, he kept an unwashed the laboratory vessel. The next day, when he observed the unwashed laboratory vessel, he found the growth of some green mold on it. Green mold. Actually, where the green mold were grown, actually he found the missing of the bacteria. Actually, he was working on the bacteria called Staphylococcus bacteria. He was working on the bacteria called Staphylococcus bacteria. I can say that Alexander Fleming accidentally discovered this medicine. It accidentally happened incident because he left the laboratory vessel, and it was containing some Staphylococcus bacteria. But next day he found this the uncovered laboratory vessel. This Staphylococcus were missing, but only green mold were grown on it. And where the green mold were grown, there was no these Staphylococcus bacteria. And later he found the green mold produced a chemical called penicillin. The chemical called penicillin. So the first antibiotic, that is in the year 1926, discovered by Alexander Fleming, that is penicillin. Later, the penicillin extracted from the two different types of fungus. One is called Penicillium notatum. Another one is called Penicillium chrysogenum. Penicillium chrysogenum. So these are now widely, these two yeast or two fungus are widely used for the preparation of the penicillin. The penicillin is a bactericidal antibiotic because it kills the bacteria, right? And also the penicillin is also called drugs. There is a queen of drugs. It's also called queen of drugs. Penicillin is also called queen of drugs. But the penicillin, it is used during the World War II by the two scientists. One is called Ernest Chain. Another one is called Florey. The two scientists, they widely used to treat the wounded soldiers during World War II, and they have found the potential effect of this penicillin medicine. So for this research, Alexander Fleming, this Ernest Chain and Florey, these three scientists were awarded Nobel Prize in the year 1945. The year 1945, the three scientists awarded Nobel Prize for this great discovery. So later, after this, is the medicines like antibiotics like there is streptomycin, tetracycline, neomycin, like different types of the antibiotics where there is prepared. And now we are using all these medicines in pharmaceutical industry to cure the different types of the diseases. So the medicines like tetracycline and chlorotetracycline, streptomycin. These three are called there is a broad spectrum antibiotics, and tetracycline is the bacteriostatic antibiotics. As said earlier, this bacteriostatic antibiotics generally inhibits the cell wall synthesis of bacteria, and also inhibits the protein synthesis of bacteria. And in a tetracycline group, the chlorotetracycline was the first that is antibiotic that is prepared it is actually prepared by using these microbes called it is a Streptomyces aurefaciens. Name of the bacteria called microbes called Streptomycin aurefaciens. It is a fungus. And streptomycin is obtained from the fungus called Streptomyces griseus. Streptomycin is obtained from Streptomyces griseus by the scientist called Selman Waxman. That is in the year 1943. This medicine is useful to treat the disease like tuberculosis. Tuberculosis caused by Mycobacterium tuberculosis. So Mycobacterium tuberculosis that can be treated by using the medicine called streptomycin, and other microorganisms, other the product, that is antibiotic product like erythromycin, neomycin, chloromycetin, grisofulvin, and neomycins. Like these are also, there is a product that is produced from other microorganisms. All these are the potential antibiotic substances that is produced in the various industries. Okay, the remaining industrial products like beverages, vaccine production, pharmaceutical the drugs, enzyme synthesis, all these things we'll discuss in other classes. So I think all will watch these videos, and in addition to that you go through the book also. Thank you very much.